Let us see the chemiosmotic theory where ATP is produced. Okay. So to consume our time, we have just drawn this picture already. Okay, now this is chemiosmotic theory. Now see here. So this is one thylakoid where it consists of thylakoid membrane. Right, this is thylakoid membrane. Just now we just gone through the electron transport of non-cyclic reaction, right? From photosystem 2 to photosystem 1, electron is going to transport where it is producing NADP H, that is reducing power for synthesis of carbon dioxide, carbohydrates, right? Now the whole system again I just written here to understand how the proton gradient occurs in the lumen side, right? Now what happened? Photosystem 2, when gets maximum incident of light, that is 680 nanometer, it becomes excited, okay, and electrons moves from photosystem 2 accepted by primary electron acceptor where it transports through electron transport chain, right? Plastoquinone, plastoquinone complex, and through that cytochrome complex where cytochrome B6 and F is there, through that plastocyanin. Okay, now whenever the electron is moved from plastoquinone to cytochrome complex, then a pair of proton is accumulated inside the lumen, right? Now here, to continue the cycle, the last electrons are provided by the water splitting complex, right? Where it releases, by splitting of water, it releases a pair of electron, right? This pair of electron again goes to photosystem 2 to continue the electron transport chain, right? Now, along with electrons, protons are also released inside the lumen, right? Now here the concentration of protons increases in the lumen where the concentration of protons decreases because already through quinone cycle a pair of protons released into the lumen, right? Now to produce the reducing power, it also requires a pair of proton which is intaken by NADP reductase from stroma, right? Now what happened? By this, the concentration of protons in the stroma is decreases and concentration in the lumen increases, right? Now here the concentration is more, so that's why protons tends to move from higher concentration to lower concentration, that is from lumen to stroma side, right? But protons cannot move directly from lumen to stroma because there is a membrane is there, thylakoid membrane is there, right? So to move from higher concentration to lower concentration, it requires a proton gradient channel where F0 and F1 are acting as proton gradients, right? Now, so here pair of protons is moving through F0 channel, it release energy, right? So it release energy, protons release energy, that energy was used by ATP synthase enzyme, that is F1. Here ATP synthase enzyme is there, right? So what is the role of ATP synthase? It intake adenosine diphosphate along with inorganic phosphate to produce ATP, right? Now see here, while moving protons from F0 channel, okay, F0 channel, it release some energy. It release some energy. While protons is F0 channel is releasing energy, ATP synthase utilizes that energy to form adenosine triphosphate from adenosine diphosphate and inorganic phosphate. Now, after producing ATP, again the pair of protons are released into the stroma. Now, from lumen, the protons are moved towards the stroma and the concentration gets balanced. Like this, our ATP is produced, right? Which is the end product of non-cyclic electron transport. Now this ATP is released throughout the stroma, okay, and NADPH, that is reducing power, is also produced inside the stroma. Right. Now this ATP and NADPH is used by the plant to prepare carbohydrates, that is undergoing biosynthetic phase of, that is dark reaction, right? So dark reaction is taking place in the stroma. Until now we discussed the light reaction where ATP and NADPH are produced and oxygen is also released outside the atmosphere as a byproduct, right? So this is about non-cyclic photophosphorylation. This chemiosmotic theory where ATP is produced. Okay, so this is clear.